Hello, my name is Bhismadev Chakrabarti. I'm a professor of neuroscience and mental health at the University of Reading in England. And I'm also a visiting professor of psychology at Ashoka University in India. And what brings me to this conference is I'm also the research director of the India Autism Center that's coming up in Kolkata. I'm very happy to talk to all of you at the BIMSSCON this year. I would have preferred to do this talk in real life, but here we are. And I'm going to talk to you about the experiences over the last decade or so that we have had in moving our lab-based neuroscience research to the field. And the area that we are focusing on is autism. And I hope that you will find some of these experiences informative. I'm going to share my screen now, so let us hope these technical challenges do not bother us through the rest of the talk. Here we are. Okay. So the topic of my talk is moving the lab to the field, perspectives from autism research. In mapping this journey, it starts off at the laboratory. So I trained as a neuroscientist. So most of our work started and continues in the laboratory. Over time, some of this work has moved to the field. And when I say field, I mean in outside the laboratory, in schools, in home settings, in community settings. And this translation to the field is aided further by technology. So this move towards technology has come in only in very recent years. And finally, just to complete the loop, the data that we are gathering from using all these various kinds of technology are again feeding back into lab-based research. So this continuous transition between the lab and the field is very much the direction of the future when it comes to autism research, or really when it comes to any kind of neuroscience and mental health related research. Because the days of just studying 10 or 20 patients and 10 or 20 controls within the lab setting, those days are numbered. We have to move to a bigger data. We have to make more robust inferences from large-scale population studies, and that's where this journey becomes informative and interesting. When we talk about topics, what kind of topics do we cover here? We start off with psychology, neuroscience, the blend of those two topics, because that's the starting point. But very soon we ventured into epidemiology. And from epidemiology, we had to venture into what's now called digital health or mobile health technologies. And all of that, the output from that mobile health technologies is feeding back into neuroscience research again. So that is the loop in terms of fields. So when one thinks about specialization, which is one of the common things that many students like yourselves would be thinking about, while it is very important to specialize in one or the other discipline, if you're specializing in public health or psychiatry or neurology, it might equally well be worthwhile thinking about how these things tie up with other specializations, because it's unlikely that even if you specialize in one domain, that's the one specialization that will stay with you through life. I mean, I trained as a neuroscientist, but over years I've had to learn epidemiology. Over years I've had to learn the digitization of health technologies. And so you start growing into different specializations from the one that you started from. But I hope that just hearing about this journey that I'm going to take you through in the next 40 to 50 minutes will give you an idea of how you can start in one place and end up in another as you go through and target real problems that face us and our countries in this time. So very first thing is starting point, which is the starting point of neuroscience or psychology. And I have been very interested in autism. Those of you who are not familiar with autism, it is a set of conditions that are associated with difficulties in social behavior in particular, but also repetitive behavior and restricted range of interests are seen in many of these individuals. It is a lifelong condition, and the individuals affected by autism spectrum conditions show a variety of abilities. So you can have a very severely affected person who needs a lot of support. As well, you can have individuals who are doing wonderful things in everyday life and really very high achieving individuals, but they also have the condition. So you have a whole spectrum of abilities and features that are represented within this community. But one thing that is seen in most individuals with autism is certain kinds of difficulties in making sense of other people's emotions or mental states. Many of us understand other people's emotions or mental states almost automatically. We see another person's facial expression and we can guess what emotion they are feeling. We hear a person's voice and we know what emotion they must be communicating. But we often don't think about how does the brain actually go about doing this? This is not a straightforward problem. I mean, it's if you think about it, it's just a set of contorted facial muscles or certain kind of packets of air coming out from the mouth that is giving rise to this very complex signal. But we always interpret this as, okay, this person must be happy or this person must be angry. One of the hypotheses that has been put forward for autism is that individuals with autism often do not have as much of a preference for social stimuli. So just as many of us would naturally find looking at other people to be a rewarding phenomenon, we would enjoy looking at, interacting with other individuals in our day-to-day life, many individuals with autism do not find that to be a rewarding experience. This is the starting point of the social motivation hypothesis in autism. According to this hypothesis, if a child right at birth does not find another human being or another human face or voice to be rewarding, they would not attend to that person. So reduced preference for social stimuli would almost automatically lend itself to reduced attention towards social stimuli. Reduced attention towards social stimuli would end up resulting in reduced learning from social cues, because a lot of information that we learn as human beings in early development is by focusing on our parents or caregivers and seeing what they're telling us or copying their actions. But if I'm not interested in social stimuli to begin with, I miss out on those learning opportunities. And that is why these learning opportunities, if you miss out on learning of social cues, and that goes on for years and years, you end up being a person who does not understand social cues that well because you did not attend to them that well in the growing up years. So which can then be interpreted or manifest as what we call atypical empathy. So the reason why an individual with autism might struggle to make sense of another person's emotion expressions might have to do with the fact that there is an atypical reward responsivity to social stimuli, by which I mean that they are not rewarded by social stimuli as much as non-autistic individuals. So the first set of studies that we started doing in this area was on quantifying social reward responsivity in autism. Now, social reward responsivity is a term that we use to really, it's just a kind of more technical way of talking about how much reward response do we see in autistic individuals when they are presented with a social reward stimuli, like a smiling face here, compared to a non-social reward stimuli, like a bunch of cakes. And in this set of experiments, and we did a bunch of experiments which are very similar on this lines, so we present social and non-social reward stimuli next to each other on a computer screen while we are calculating how long people look at the social reward versus the non-social reward using an eye tracker. For those of you who are not familiar with eye trackers, the eye trackers work using a simple phenomenon of throwing an infrared light from the eye tracker to the pupil and then mapping the reflection of that infrared light on the screen, and then it gives us a very clear estimate of where exactly a person is looking and for how long. And what we found was that individuals who were high in trait empathy, which is measured using EQ, which is shown in the x-axis of the graph here, individuals who are high in trait empathy looked much longer at the social rewards, the smiling faces, compared to the cakes, which are the non-social rewards. Whereas individuals in low trait empathy do not really show a preference for social or non-social rewards as such. So they're much more on the fence. And the relation between empathy and autism, as I said before, individuals with autism usually score low in trait measures of empathy. So here we have an observation that individuals who have low trait empathy, such as those with with an autism spectrum condition, do not really show the social reward preference that we see in the high empathy individuals. We followed this up with another experiment inside the scanner. This is an fMRI scanner. This is an MRI scanner, and what we have done is a functional magnetic resonance imaging experiment where we are presenting social and non-social rewards one at a time to individuals with and without autism. And what we see is and what we are measuring is the brain activity in a reward processing region, which is the medial orbitofrontal cortex, and how that connects to the face-related region. So we are measuring the coupling between a reward processing region, which is the medial orbitofrontal cortex, and how strongly is it coupled to a face processing region. So we are literally trying to work out the social reward circuit here. And what we find is that the strength of coupling between this face processing region in the fusiform gyrus and the reward processing region in the medial orbitofrontal cortex, that coupling is weaker in individuals who score high in autistic traits, here measured using AQ, which stands for Autism Spectrum Quotient. So if you look at this right-hand plot, you would see that individuals high in autistic traits have a weaker coupling between the brain regions involved in processing rewards and the brain regions involved in processing faces. these experiments that I just talked about in the previous slide were done on adults. We have then gone on to do measures of social reward processing in children, very young children, toddlers with and without autism spectrum disorders. And we designed the first kind of mobile experiment at that time for doing so. This is back in 2016, 2017, when this was designed as an app, very small app. On an iPad, you would see two bouncing balls, basically. One of them is a red bouncing ball and another is a blue bouncing ball. And if you press the red ball, you see the picture of another child coming up. If you press the blue ball, you see the picture of a train coming up. And what we are essentially measuring is how many times does a child with autism press the red ball versus the blue ball. So if you prefer the social stimuli more, you'll press the red ball more. If you prefer the non-social stimuli more, you'll press the blue ball more. And what we found, so if you look at the right-hand plot, the part of the plot that says real, what that shows is that autistic children press the social, the ball associated with the social stimuli, the red ball here, far less times than typically developing children. So typically developing children press the social button more compared to autistic children, which is very similar to the results that we saw from the adult data that I showed in the previous slide. We wanted to check, though, that whether this preference that we are seeing here with button press, whether that was due to the fact that there is something about the contrast of these images. So low-level visual properties like contrast or color or complexity of these images that might drive this, this preference. So what we did, we created scrambled versions of these images as well. So we scrambled the child face, we also scrambled the train image. And at that time, we set it up as a separate experiment where the red button was associated with the scrambled version of this child image so that you can't tell that it's a child, but all the low-level properties of that image are preserved. So the contrast and stuff is all preserved. And the blue button was associated with the scrambled version of the train image. And we find that once you scramble the images, then there is no significant difference in preferring one image over the other or one button over the other. So that told us that what we are seeing here is not really explained by the low-level visual features, but it is indeed a case of autistic children, and these were 14-month-old to 40-month-old children. These very young children, they genuinely did not prefer the social rewards like smiling baby faces as much as they did the non-social reward. This was done in very young children using a tablet. We come back to discussing another paradigm in mapping this social reward responsivity in autism. Back to adults. And this technique is a more complex technique, something called continuous flash suppression. So what I've been talking about so far is techniques, both, so I've talked about an eye tracking study in adults, I've talked about an fMRI study in adults, and I have talked about a tablet-based study in toddlers. All of these cases, all of these three studies, we presented the stimuli above the threshold of awareness. So the children or the adults had all the time in the world to look at the stimuli that are presented to them and then make their decision on pressing a button or not, or decide where to look, whether to look at the smiling face or the chocolate, as the case might be. But what happens if the stimuli are presented below the level of awareness. This is where this new technique comes in called continuous flash suppression. Now, I am not sure whether the visual is very clear because it is very, the contrast is very low on these stimuli, but I'll try and explain. So in this set of experiments, what happens is you present two different screens to two eyes, and you put a partition right along the nose so that the right eye cannot see what's there on the left visual hemifield, and the left eye cannot see what's there in the right visual hemifield. And what we do is we present a video almost of jumping ellipses, very high contrast ellipses. So what you can see in the left eye in this slide here. And on the right eye, we present a static picture of a social reward or a non-social reward. Now, the phenomenon, why it's called continuous flash suppression or CFS, is because if one eye has a continuous dynamic pattern like these jumping ellipses that I've talked about, so these are kind of flashing ellipses at 10 hertz being presented to the left eye, then people actually cannot see what is being presented to the right eye for a while. So it is several seconds after which they will say, well, oh yeah, I did see something on the right eye. And then we ask them what they saw, and so we know that they are not lying, basically. And this whole idea that presenting dynamic stimuli to one eye and a static stimuli to another eye makes the static stimulus invisible for a while because of the nature of our visual system. This phenomenon is called continuous flash suppression. And what we found was that autistic adults did not show any advantage in detecting social stimuli or non-social stimuli. So if you look at the bottom plot here and focus on the bars that say normal, that shows how much time did autistic adults take to detect the static social stimuli, which is just around 3 seconds mark. And the gray bar shows how much time did autistic adults take to detect the non-social static stimuli presented to the right eye. And you can see they are very similar times. So there is no difference between how quickly they detect a social versus a non-social stimulus. In contrast, now look at the top set of bars here and the bit that says normal. You'll see that the social stimuli are detected much earlier than non-social stimuli in typical adults. So the top set of bars are from the data from typical adults, and they detect social stimuli much quicker. The white bar is smaller than the gray bar. They're showing that even below awareness, autistic adults do not have the advantage that typical adults have in being able to detect social stimuli quickly enough. So this kind of brings to a close the neuroscience part of some of the stuff that we have been interested in. So this is something we are we have been studying at social reward processing in autism using a diverse set of experimental techniques that include continuous flash suppression, eye tracking, fMRI, and tablet-based measures. Now, at this point, this is a standard fare autism neuroscience track of work. At this point, I'm going to take a jump into epidemiology, and I'll tell you how that came about. So, all the time that I've been kind of working on developing these neuroscience-based studies of social reward in autism, every time I would go back home to India, I would be asked a very simple question that how many children in India have autism? And I would usually say, well, you know, we have the American figures and we have the British figures, but there hasn't been a single population level prevalence study of autism in India. So, and that that's always been an area of discomfort for myself. That, you know, I work on autism, and yet we do not really know how many children have autism in India. And so what I have here is the world map as of 2012, and things have moved on since then. So if you want to see a more up-to-date map of autism prevalence estimates across the world, you should look at this web link that is provided here. But this is almost a semi-historical account of how this started, that all the numbers that you see here, that 16 in Europe, 12 in Japan and China, 8 in USA and Canada, these are all number of prevalence studies of autism that were there in 2012 that were published, and India had 0. So, and why do we need to know the prevalence? Why do we need to detect autism? Because if we know the numbers, then we can. That's the first step towards establishing what kind of needs are there, what kind of services need to be in place. And so it's the first step to designing services and getting an idea of the need is really to do an estimate of the prevalence. And early detection is absolutely vital for better outcomes. So this is something that has been talked about as a detection barrier, because we know that better and early detection of autism leads to better and early intervention, and better and early intervention of autism leads to better outcomes, because the brain is very plastic in the initial years of life. If somebody has to wait 10, 12 years before they are diagnosed properly or intervened properly, then the chances of improving are far lower than if they have been diagnosed in the first couple of years or 2.5 years of life. So early detection and early intervention are absolutely vital in terms of improving early outcomes. And there are a lot of barriers that you would be already aware of. And the first one that I came to realize when I thought that, okay, we will do a prevalence study of autism, is that the tools, the screening and diagnostic tools, were just not available in the local languages. Everything is in English, and that just wouldn't work. That we can't really work with English tools in India across all socioeconomic segments. There are also other existing barriers for early detection. There are very few clinical experts in mental health. So as of a few years ago, there were a total of 3,000 mental health professionals in a country of 1.2 billion in India. I do not know the number in Bangladesh, but I would imagine that it's not terribly different. So our countries are very much suffering from this dearth of clinical expertise in mental health. And even if whatever expertise there is, they are concentrated primarily in cities. So you move out of cities, and then the needs are even greater. Plus, there is a huge variability in culture, language, socioeconomic status, and coupled with all of that is the low public awareness and stigma of mental illness. So the whole idea that the one word, pagol, captures the whole spectrum of mental illness, that in itself is a huge thing to fight, because mental health has so many diverse categories within there, and as I said, a diverse range of abilities are represented there. So we have to start somewhere. So this is where we started. And we started with translating and validating the autism screening and diagnostic tools to local languages. And in India, our first choice, being Bengali myself, I chose Bengali, and I started working in Kolkata. But because we also had connections in Delhi, so we had Hindi as the second language that we chose to focus on. Together, Bengali and Hindi have about 500 million speakers. So we are talking about a sizable number of people in the world who so far had not got access to autism screening and diagnostic tools. So we started with the kind of very basic work of translating. So the way it works is you translate an English tool to Bengali, and also you change things that are culturally suitable. So, sometimes there are references to Christmas parties or birthday parties in the Western affluent society-based tools, and we had to change them accordingly. And also we have to change references to certain children's games to make sure that these are compatible. And then once we translate them to Bengali, then we get another independent person to translate them back to English. This is to make sure that the back translation is not biased. We do the translation, but we do not get involved in back translation, and that so that we know how is it being perceived by somebody who doesn't know this field at all. And then the back translation is compared with the original version of the tool with the person who designed the tool, and then we go through this iterative loop multiple times. So this is translation and back translation going on. So we chose a bunch of tools that are listed on this slide: Social Communication Disorder Checklist, the Autism Spectrum Quotient, Social Communication Questionnaire, and the ADOS, the Autism Diagnostic Observation Schedule. These are all screening and diagnostic tools. In addition, to build the kind of research toolkit for autism researchers in South Asia, we also made Bengali translations of standard cognitive behavioral tasks that have been used in autism research. Some of these are emotion recognition tasks, embedded figures tasks, as well as reading the mind in the eyes task. I'm happy to go over any of these tasks in detail at a later point if anyone is interested. And all the papers are cited here, also from our lab website that you can see on the bottom of the slide. So here was the first set of results that we found from our translations. What we wanted to check was how do autistic children, perform in these different tools. So what you see here is the mean score that is received by autistic children on each of these tools using the original tools. So this is the original would be in English. So the mean score when people use an English version of the AQ is just around 100 in the British sample. And in the sample that we got from Calcutta and Delhi, the Bengali version is around 85 mean score. The Hindi version is around 95. And similarly, you see that all these mean scores from autistic children using different versions of these tools are very comparable. So that is a very kind of quick proof of principle that these tools are functioning roughly in a similar manner. In the next few slides, I'm going to talk a little bit about how we take a slightly deeper look into these tools and cultural differences. But before that, let me highlight one of the insights that we got with using these translated versions of these tools. So one of the things that we discovered and that we did not expect was that it made a big difference on who was filling in the questionnaire. So for example, the SCDC, or the Social Communication Disorder Checklist, if the parents were filling in the questionnaire, this was in Kolkata, almost 20% or 1/5 of the children met cutoff for autism. So this cannot be right. So that's something is not quite right. If 1/5 of the entire set of 6,000, almost 6,400 children are meeting cutoff for autism, that possibly means that parents are over-reporting. In contrast, when we look at teacher report on that same set of children, we see that only about 4%, 3.6% to be precise, met cutoff for autism. So that would mean that teachers are using a slightly different set of criteria to report on how many children meet cutoff for autism. And that is possibly because teachers do see a much wider number of children than parents do. That might explain why we have a huge case of over-reporting in parents compared to teachers. The similar pattern was seen also in Delhi, with 11.5% parents, 11.5% children meeting cutoff for autism in parent report, whereas only 6.7% of children meeting cutoff for autism on teacher report. And here is another methodological insight that might be worth keeping in mind. If we wanted to see whether parent report and teacher report was correlated, and if one focuses just on p-values for identifying significant correlations, you will see both of these have very significant correlations, less than 0.001. P-values are 0.001. However, the key thing here, if you look at the plots, the R values are very low, 0.06, 0.11, and the lines, these regression lines, are almost flat, which would mean that the correlation doesn't really exist as anything meaningful. It's a very weak correlation, and the only reason why we get such a low p-value and this statistical significance is because we have a huge sample. So this is something that's worth keeping in mind when one is looking at just the statistics of the data. It's always good to look at the plots of the data and really figure out that is there indeed a relation between parent report and teacher report? That basically means that do the same children get picked up as high autistic traits by the children, by the parents and the teachers? And the answer, if you were just focusing on the numbers here, you would think that yes, the same children are getting picked up. Actually, it's fairly random on the children who are getting picked up by teacher report versus the children that are getting picked up by parent report. So inter-observation correlation is very low. So the key thing is to focus on the effect size and not just the p-value. So that was a methodological insight. Now, moving on, here is the point that I just referred to in one of the previous slides, in kind of taking a deeper look at the cultural differences aspect. In looking at cultural differences, what we looked at was to combine our data on the autism spectrum quotient, the AQ, from 3 different cultures, from India, Japan, and UK. And what we found was that the analysis is something called an item response theory based analysis, where we take each item of the autism spectrum quotient and see whether it discriminates well between autistic and non-autistic individuals. And what we found was very interesting, that all the items that you see on the top box here, that starts with can easily keep track of different conversations, enjoy social chit chat, and so on and so forth, all of these items were universal. That would be that all of these items were very good in discriminating autistic from non-autistic individuals in all of these 3 cultures. Whereas if you looked at the items in the gray box, these ones are very different culturally. So when talking, it is not easy to get a word in edgeways. This is a very good item for discriminating between autism and non-autism in UK, because in general, I guess the social norms are that people wait until the other person has finished talking and then start talking. Whereas in India, it did not discriminate between autistic and non-autistic people at all, because in general, this is a speculation that we have put in the paper. It is perfectly acceptable in India to almost talk over another person. So somebody, you don't wait until the person finishes speaking and you start speaking yourself. And that's why the discriminability of that item in being able to separate autism from non-autism is not very high in India. Similarly, questions, items such as enjoys doing things spontaneously works very well in UK, but doesn't work in Japan, because of possibly a slightly more structured society than what we have in the UK. So there are very interesting insights to be had by combining data sets from across multiple cultures. We would know, I mean, we start thinking that, just because we have made a translation, it will work exactly the same way. It doesn't. There are certain items that work better in certain cultures and other items that work better in other cultures. So this is worth keeping in mind when one thinks about designing studies that go across multiple cultures. In my next slide, I just present a little bit of the data from the performance-based tasks. So here, the tasks, if you look at the plots C, D, and E, those are the data from autistic children in blue and control children in red on tasks that involve emotion recognition. This is plot number C, recognition of the eye region of the face to infer the emotion. In plot D, the task is called reading the mind in the eyes task. And finally, an embedded figures task where you have to discriminate embedded patterns from more complex visual display. And again, what you see is that the groups between autism and control, the blue and the red, are very clearly discriminated. What this paper in 2016 we showed was that these tasks work very well in Bengali-speaking communities, and just as they have been shown to work in the West. So the performance measures are very clear in achieving a group separation. Now, all of these tools that we developed using the translation and back translation, the Bengali and Hindi tools, we then used these tools to conduct the first prevalence study of autism in Indian school children. So we focused on 28 schools in Calcutta, and we collected parent report and teacher report on them. Then those who screened positive on the SCDC, this is the quick screener, we then tested them with social communication questionnaire, SCQ, as a second level validation. And those who screened positive with SCQ then were given the ADOS, or the Autism Diagnostic Observation Schedule. And then we calculated the prevalence, which was around 0.23% in the sample that we studied. And we really went to all different socioeconomic segments. We went to private schools for the high socioeconomic segment, as well as Anganwadi schools for the unorganized sector. Now, all of this work, when we were conducting the prevalence study, was done using paper and pencil questionnaires. That work takes a long time. So for us, it took about 5 years. So there is a need to move to technologies that scale. And that's where I come to the final part of my talk, which is on mobile health. So moving beyond paper and pencil-based questionnaires, we tried to create an app that would have measures such as questionnaires, but also performance-based measures like the lab-based social reward paradigms that I've talked about earlier in the talk, all in one platform that can be administered by non-specialists in the wider population. So this was the first project that we did called START, which stood for Screening Tools for Autism Risk Using Technology. And we were partnered by a whole bunch of groups across the world, and you can see some of those institutions named here. And those of you who are interested in this project, you can look at the website there. And the aim of the START project was to develop a mobile, modular, open source platform for early detection of autism risk by non-specialist health workers in low resource settings. Now, you remember the prevalence study that I just mentioned. There we had to have clinical psychologists going and testing these thousands of children, essentially one at a time or in small groups. That in itself is a very slow process and it does not scale up, which is why we need a non-specialist, not a clinical psychology specialist. A non-specialist should be able to administer it on a tablet, and the data from the tablet should be able to go to be uploaded to a central server from which we can download and analyze. And the START project had questionnaire measures like the ones that I've talked about in translation, but it also has task measures, which are similar to some of the ones that I've talked about in the first part of the talk in the lab-based work. This is exactly bringing the lab-based experiments and translating them to a mobile format. And that's where, so the social preference or social motivation measures would be, I'll give you an example of how we are doing it in the START app. We also had measures of attentional disengagement, sensory preference. So how many autistic children have preferences for looking at repetitive stimuli or stimuli that are rotating and have a certain pattern. So those tasks have all been incorporated within the START app, as have been some tasks of motor performance and visual motor coordination. Finally, so this is a little bit more detail about the questionnaire measures that we have used, which are similar to the ones that I've talked about already. And the most interesting thing about these questionnaires as administered within the START is that they don't need to be just based on text. So, for example, one of the things that we realized while doing the prevalence study was that sometimes some parents would interpret words differently. So they would not really know what we really mean by saying, oh, is your child doing things spontaneously or not? Whereas if we could show parents a choice between one video where a child is behaving in a certain way and another video where the child is behaving in a different way, in that case, and ask them to say, well, which child within these two videos is your child more similar to? That way, it's much clearer a thing for parents. So this is a functionality that we've built into the START app, that we can design video-based questionnaires rather having to rely on text-based ones. And then we had a big kind of technological innovation that we have built in there as well, which is eye tracking using the front camera of the tablet. Now, you would remember in one of my very first slides, I talked about how we were presenting a smiling face next to cakes. And we were measuring using an eye tracker, how much time do you look at the face versus the cakes. That's done using a very costly infrared eye tracker. Whereas what we have done here is to use the front camera of the tablet to record the eye position. And from the movement of the pupil, we can guess to almost 97% accuracy if the person is looking at the left side of the screen, which is in this case the face of the child, or the right side of the screen, which is the train. And so we don't need to have a very costly eye tracker. We can get that data right from the front camera of the tablet. And I'll give you a quick video to show how it actually appears in relation to the tablet. So this is this is the other task which I've already talked about for measuring social motivation. So you'll see, you press one button and you see a non-social stimuli, and you press another button and you'll see a social stimuli. And here what we are measuring is how much how many times are you pressing the social button or the non-social button? So we have the eye tracking part, which is looking at whether you're looking longer at a social versus a non-social stimuli when they're presented simultaneously. Then we have this button task part where we are looking at how many times you're pressing the green button versus the orange button. Then we have a measure of visual motor coordination, where we ask the child to follow a B across the screen, and what we are measuring is the spatiotemporal errors in following a dynamic trajectory. The idea is that many autistic children have motor difficulties, particularly in, some of these have been attributed to cerebellar atypicalities. So, and it's one of those domains that's not widely studied in autism. So it is something that we wanted to bring in here, that the errors that one does while following this flying bee across the screen, those constitute an important measure of motor abilities. So we have measures of social performance, motor abilities, sensory abilities, and we also collect data from the free play of mother and child, or a caregiver and child. So how do they, and that we collect, so the community health worker usually holds the tablet to record video of mother and child playing for 10 minutes, and we code the different types of behavior in there to look at ecologically valid real world behavior. We have tested about 120 children with autism spectrum disorders, intellectual disability, and typically developing children in and around Delhi with this app. All the testing was done in at home settings. And we find that most tasks, we get over 80% completion in all of these children. So these are feasible and acceptable assessments and we get for eye tracking, we get over 92% data, and it is very quick to administer, which is a big thing. And this is really a proof of the pudding in terms of taking a lab-based measure outside the lab and going to the field and going to people's homes and figuring out whether we can get meaningful data out of it or not. And the main reason for incomplete tasks, which we had about 20% of the time at max, was due to child factors like loss of interest. And based on this pilot data on about 120 children, which we are about to submit for publication now, we see almost all of the tasks do very well in discriminating between children with and without autism. Interestingly though, the children with autism and children with intellectual disability, they were not very clearly distinguished by these tasks. And our speculation on that is because all the autistic children in our sample here also had comorbid intellectual disability. So these two were not very clearly discriminable groups phenotypically anyway. But what is crucial to take home here is that we could, with 97% accuracy, we could discriminate between typical and atypical neurodevelopment using this app, the START app. So here are a few numbers in terms of START app. So we made about 115 in about 1.5 years. We won a couple of prizes internationally on e-health awards. There is an annual international e-health competition in France. We got the award for the jury's award across all categories for the most innovative app at that point. And we've tested well over 400 children really now. And the app is now available in 11 languages, including Bengali, Hindi, and a whole bunch of other languages. This is an open source app, so we are always looking to forming collaborations so that this app gets used more widely. In the very final couple of slides, I just want to say where we are headed now. So our START project has finished, but now we have teamed up with other tools that are operating in this zone, this early child development zone, that measure cognitive abilities as well as these social and motor abilities. in the entire age range of 0 to 6 years. And we are going to test this combined tool, START, and these two other tools that we are teaming up with. One is called DEEP and the other is called Indigo. We are going to create one single platform that we are calling Scalable Transdiagnostic Early Assessment of Mental Health, or STREAM. So we are creating this STREAM platform that we will use in 4,000 children in India and in Malawi to create developmental norms for each of these domains. Just like we have growth norms for height and weight as children are growing, we are trying to create developmental norms in each of these domains using this combined assessment platform that we are building. So this is work very much in progress, and we have this project will run until 2024 or 2025. So in my very final slide, I just want to say that it's very crucial to move the lab to the field and also bring the field data back to the lab, because this is a continuous dialogue. And we know it's very important to have culturally validated or culturally agnostic tools in some ways. The questionnaire tools are great, but they need to be very carefully administered and translated, because words are more culturally influenced than possibly things that do not require words. Something like tracing a B across the screen, there is not too much cultural differences in there, but certain words such as, the cultural differences that I highlighted when I was talking about the study between India, UK, and Japan, those things are possibly more tricky when it comes to questionnaire-based data. So we must have a mix of culturally validated questionnaire tools as well as culture-agnostic performance-based measures. And we definitely need to move beyond just studying an autism group or an intellectual disability group and move to a larger population-based study. And to do that, we need scalable technology. Finally, I just want to thank all the people that have worked with me and my current collaborators and the funders. If you want to know more about this research or any of this work that I've talked about today, please visit our lab website, www.bhismalab.org, where you'll find all the papers and also summary videos that you might be interested in. Thank you again for your interest, and have a very great rest of the conference. Thanks.